Kempermann, and now we're moving on to how lifestyle shapes brain in healthy aging and disease. And Gerd is a professor at the Center for Regenerative Therapies, Dresden, and also speaker at the German Center for Neurodegenerative Disease, Dresden. So please, go for it.
Thank you. Thank you very much. Yeah, I'm very pleased to be here. Thank you for the invitation. I would like to tell you about the basic principles that we're trying to study in rodents, and that might help us to understand some of the issues that are discussed here. At least I would hope so. So, in general, I'm interested in the half-full glass in the context of neurodegeneration, trying to understand what it is in our brain, and perhaps in our lives, that works against the impact of neurodegeneration. And I've gone through a long list of factors that is in the literature and for which some evidence exists. So this is not rigid evidence, good evidence in the scientific sense, but good evidence in the sense that there are studies that we would call scientific on these studies. And you can see that it's about a list of 55 lifestyle factors that have some impact on prevention of neurodegeneration, improved cognitive function. And that's much more than what we normally see. I have a problem here. Let me go back here.
I may be useless, but I'll give it a try.
There's a problem here. So, no, it's okay. I started the wrong presentation, so. No, it's still the wrong one. There's a problem here. That's a new one. Right?
Yeah, that one is on you. I don't take responsibility.
Yeah. Okay. So I'm sorry. I noticed that there was something missing that I wanted to tell you about. So now we are back. So this is again where I wanted to hook up with what Lars was saying. This is from the Einstein Accord, and it tells you, cut a long story short, that there must be something that is not genetic that affects, in this case, within age cohorts, the decline in prevalence of Alzheimer's disease, in this case. So there must be something changing. And as this is not genetic, it must be related to behavior. And the WHO has recently issued these guidelines, recommendation for the first time, giving guidelines, well, this is what you should do. And the result, I mean, you've probably seen this, it's really surprising what they came up with. Don't drink too much, exercise, don't smoke, manage blood pressure, maintain a healthy weight, eat a balanced diet. Wow, this is really something. This is new, this blows. Yeah. And the question is, well, we have all known this. You can read that in any paper that you open, any tabloid, anything that you find in the pharmacy store. We have known that, but little has changed. And we know these factors are important, but we also know that they are not applied for some reason, that they might work, but we cannot improve on that use already. And this is where this figure comes in. There's many more factors that don't make it, for which some evidence, as I said, exists, but people probably wouldn't say that these 6 factors from the WHO or the related factors would describe lifestyle. You would not like to have your lifestyle reduced to 6 big effect size things. Lifestyle is something else. And this might describe lifestyle much better. And the logic of this is that lifestyle here relates to body and mind, to an input and an output, and you can classify all these factors according to these criteria, and you find factors in all of these. So there is a field of tension between receiving and giving, influencing, being influenced, passivity, activity, being object or subject, between mental and physical, self and social, energy, information, quantity, quality, you name it. There's a lot going on. It's much more complex than these isolated 6 factors from this big field of ideas might suggest. And one reason why it's so difficult to implement this might be that we personally value these things might be very different than the WHO or the studies suggest. Because what you can measure are things that are either very physical, how much calorie you take in, how much exercise you do. But what we value highly is our autonomy, how much we are integrated into a social structure, our purpose. So this is a very simple Maslow-type pyramid of values. And if you superimpose this on this scheme, you see that most of the factors we have evidence for are where it's measured. But what people think is really important is on the other end of the spectrum. And there we have little study evidence. But we see that from the logic of this, there must be some relation. So people behave in a way that might be consistent with all of this. So what is measured is not identical to what is done. The recommendations are usually in this corridor between education and exercise. It's not identical to what is missing, and it's also not identical to what this actually means in terms of lifestyle, which is the center of this all in this field of tension. So what I'm trying to understand with my research is really extremely reductionistic, because I think this cannot be studied. If you don't manage in your gigantic studies to do this, we must find a different way, perhaps, to come to some of these aspects. And my idea is that lifestyle is actually highly individual, and that there are many ways to roam. But at the same time, there is something in all of this which Ullmann has, interestingly enough, we didn't talk about this before, already alluded to. And this is this observation of communality between these factors. This is from this Norton paper on, how many are these? 6 or 7 factors again. The prevention of Alzheimer's disease. And you can see in the right column the communality of these factors. It's gigantic. And all these factors, I mean, you could, if this would be a cancer meeting, you would probably have the same factors up there. If it would be on longevity, you have the same factors. Yeah. It's always the same things. And they are good for us. That's nice. But the interesting question is, what is this communality? This G factor, if you want. And I think if there is such a G factor in here, then we will find it in a much reduced system. Then it might be a biological principle, and then we might find it. And there is an experimental paradigm in rodents which has a long history, goes back to Donald Tapp in 1949. It's called the enriched environment. There's a huge literature on this. These are the fabulous 4 from Berkeley who sort of propelled this field over many, many years till the 1980s or so. It's a very simple paradigm. You keep rodents in a larger cage, more fun, more friends, whatever. And then compare that to a control cage where it's not much fun. But you don't stress the control group. You don't do bad things to them. That they're just the normal thing, and the other get a little bit better. It's not a dramatic intervention, but this is the only intervention that you do. And the effects on the brain are massive. It's a very robust and extremely strong effect. And Marion Diamond has written a book on this paradigm, old book now, but it captures the idea of this paradigm. Enriching heredity is the idea the environment acts upon a genetic baseline. Yeah, so this is a phenotype equals genotype once environment, and you keep the genome constant because you work in inbred animals, so very straightforward. But if you think about what the environment can actually do, it does impact on us in a very complicated way through many levels, and the mechanisms by which that then ultimately might lead back to changes in behavior go through a plethora of complex nested mechanisms. So this is not linear at all. It's more like this, that you have network-like structures on all these different levels, and you have an inward causality from the world to the genes, all the way down, and reverse from your genetic background all the way out to your behavior and cultural integration, you name it. And there must be interaction at all these different levels, and perhaps between these levels in massively complex ways. So if you reduce it to very behavioristic-like input-output relationships with a black box in the middle, you're missing something. But perhaps there's a way of addressing what's going on in between. And one thing is that this old paradigm actually neglects something, because in reality, we also keep the environment constant. It's always the same environment. Tiny changes, that some toys are reshuffled, but in reality, in the enriched environment paradigm, we have genetics constant and we have the environment constant. So what happens under these circumstances? We did an interesting experiment actually with Ullmann and Andreas Bandmeier, who's here, and asked if we look in an enriched environment, what happens? So we have to work with a different cage. This is now the newest version of this. This is the Buckingham Palace style experiment. Buckingham Palace, as you know, has 750 rooms, or 75 actually. And so we had a Buckingham Palace for mice as an enriched environment. That's how it looks. It's a rack, standard rack of mouse cages, but there's a crucial difference. You can see that here in that little video where the mouse can actually pass through the next cage. So these 70 cages are all connected. And so it's one gigantic cage with the resolution of one cage of 70 individual cages. And you can see between these cages there are ring antennas, and the mice are equipped with little rice-sized transponders in their skin and the neck. And whenever a mouse passes between two cages, we get a signal, and we can know where the mouse is at any time. Yeah? And when you look at behavior over time, and Andreas was kind enough to find a very cool parameter for us, which is called roaming entropy. It's the probability that a mouse is found at a given location at a given time as a measure of territorial coverage and activity. You can see that over time the animals habituate a little bit to it, so roaming entropy goes down over 3 months, but at the same time the variance goes up. So how can that be? Hey, genes are constant, environment constant. Mice habituate to the environment, but they become more different in behavioral trajectories. This can only come from their individual behavior. There's nothing else that could explain this. Right? And in that paper that I was mentioning, we showed that this increased difference in behavioral trajectories is actually correlated to adult hippocampal neurogenesis, the production of new neurons in the adult brain. And 22% of the variance in adult neurogenesis could be explained by these relatively simple behavioral trajectories. So that means that in this classical paradigm, we actually have a source, or we have an access to looking at this non-shared environment. We can turn this paradigm into one that looks at exactly the contribution under highly constrained conditions, at exactly the contribution that the individual makes to his or her own trajectory and benefit that it might gain in that paradigm. Yeah? And this is something that you might be envious in your studies, because you can control nothing, we can control everything and still see a difference. I find this cool. Yeah. Now, if this is what I've shown you, yeah, you have this decrease. We've seen that in several studies now. You have a habituation over time and an increase in variance. Now, this is a study where we used mice that do not have adult hippocampal neurogenesis. I cannot explain now in detail how that works and why they don't have that and what not. But if you don't have adult hippocampal neurogenesis, you don't see that. So it seems that you might need your hippocampus and the plasticity in the hippocampus to show this habituation and to show an increasing divergence of behavior trajectories. And that makes sense, because the hippocampus is the place, the filter for all the information that you get from the outside world. Now, something we haven't heard about today in the studies is that there must be something that links the effects on the genome, that might link the effects of the behavior to the genome, because that's the only basis that we have that can. And so this can happen acutely by gene regulation, but there must also be long-term changes, otherwise you wouldn't see the trajectories that we see in all these studies. And that's presumably lie in epigenetics. So we used our mice and looked at epigenetic changes in the hippocampus after exposure to enriched environments. And what you find, and this is just a cluster, and it's cutting a long story short, is that all the epigenetics, not all, but most of the epigenetic changes that we find cluster in structure of the neuron and in plasticity pathways, And if we seed a curated list of genes that we know are related to adult hippocampal neurogenesis, we find the highly enriched in this domain. So it seems that epigenetics is influenced by their stimulus. So it means that midterm to long-term changes of regulation might be mediated by this. And they cluster in genes which are related to the plasticity of the structure we're looking at. Yeah? And if you now look in aging, you see loss of methylation, gene methylation, DNA methylation in several loci across the genome. And we find that many of them are counteracted by adult neurogenesis, by environmental enrichment. And again, these genes that are counteracted here in the center are again the plasticity genes I was talking about. Now, how can we move to a more general take on the brain? We have now looked at a little bit at hippocampus and at neurogenesis. What can we work with? So this is the first imaging study that we did together with Tony Brennan in King's College in London. So we used our mice enriched environment with the others in a cage with in this cage system, and we looked at what the brain looks like in general. We can first see that the hippocampus changes. That's good. So hippocampus again, even with an MRT, you can see. But now we looked at all the changes, voxel by voxel, across the entire brain, and asked gray matter change and white matter change between all these regions. How do they correlate? We looked at based on a digital atlas in, I think, 138 regions. And you can see there's a correlation matrix in the control group between all these regions. Brown is a positive association and blue is a what? There's some correlations, yeah. Not surprising, right? But now if you look at the enriched animals, you can see that they become more similar in the sense that there is a higher prediction from the higher correlation between these regions. So this was first a surprising thing because, yeah, it's new, so nobody had seen that, and it might have to do with the de-differentiation paradigm that Urman has talked about. But if we look at individual groups now, we can't really break it down to individuals because then you do not get variance anymore. So you have to look at small groups. But we can stratify the enriched environment group. Yeah? So this is what we have in all animals here. And now we stratify them by the behavioral trajectories, whether the behavioral trajectory is sort of flat or upward. There's a certain ceiling effect in these cages, so these are the flat roamers, versus the downward roamers, which habituate strongly, even in the enriched group. And the stable trajectory, they have an even stronger effect of this de-differentiation, if you will. But the others, the downward trajectory, folks, they tend towards the standard condition. So if you compare these with the standard, it looks like this. So in the enriched environment, you have a range between control and super effects, and these effects are presumably based on the individual behavior trajectories. Again, it's not yet broken down to this individual level because we can't really do that with this approach, but we can do it in the strata that we have. Yeah. So, in summary, we are interested in your biology underlying the communality of lifestyle, dependent risk and resilience, and try to find reductionist models of this. And adult neurogenesis might be one of the drivers of brain individualization that we can use in these studies as a meaningful and reasonable readout. Enriched environments prominently induce epigenetic changes that are related to plasticity and adult neurogenesis in the hippocampus, supporting this claim. And environmental enrichment individualizes connectedness or correlatedness across the brain, whatever that exactly might mean. These are the people. I would also like to mention the cooperation with Ullmann and Andreas, and I thank the mice who volunteered for the study, and you for listening. Thank you.
Thank you for that, Gerd. Any questions?
I know, went a bit over time, so.
We'll give Imre one shot.
Great talk. So this is a question you probably get a lot. So how much is actually genetically determined?
What?
In the individual variability that you see emerging?
Well, here we look, we keep the genome constant, so they're inbred. So we cannot distinguish, look at any genetic effect in this paradigm. Right? There's an interesting side note. The history of this field was that they always claim enriching heredity, but the old studies are all done in outbred animals, so they're not genetically identical at all. So it's a little irony that part of the old history is not even correct, but yeah. Okay, this is an important point. Of course, in reality and what you're looking at in humans, there's a huge genetic effect. But the point is that we control for that, and we can control for the environment to expose this non-shared environment effect, which is difficult to access in other situations.
Yeah, thanks, great talk. I just kind of come back to your first of the start of the talk, really. It's more a comment, and maybe you also just want to comment on it. What I sometimes think when you see those 7 factors, and actually that's the most important for everything, lifespan and cancer and so on. Shouldn't we maybe put actually more money into getting people to do what is already known rather than finding very intricate new ideas on what might work? Because when patients come to our stroke unit, they see those 7 factors, nobody cares, and they go out and they still don't care. And sometimes I think money would be wisely spent in trying somehow to convince people to change those factors.
Absolutely, I absolutely agree. I'm not saying that you have to adhere to all 55 in order to be successful. I'm only saying that the reason why people might not adhere is that in the big picture, people might have very different emphases and values on this, and for genetic reasons, some things might better work in one person than the other. So there might be many ways to roam. At the same time, all these studies suggest that there is this communality. So if you find the G factor in this, and if you do it with these 6 factors, great. So I'm not arguing against that. I'm only saying that this might help to explain why it's so difficult to implement the things as a finger study with a few interventions. Trying to get that out of the lab into real life is hard, and this might help to explain this. Trying to expose what is the factor that is underneath all this in order to facilitate rather to make it more complicated. Guy?
Yeah. How long do you think we need to show this epigenetic effect? Do you think that for mouse, for example, we need 1 hour training or enriched environment, or 2 months, and translate it to humans? How much do we know?
You can get epigenetic
about
changes very quickly, but the question is how they last. The shortest time point we have is 4 weeks. I don't know what has been done in humans. The problem is it's tissue specific. So if you're interested in the brain, you probably don't want to poke the brain to get to the epigenetics of the brain. So you probably would use lymphocytes, which most people use. And that's, of course, interesting enough, but it would be interesting to know how well the lymphocyte predicts the brain. But I think we need to do this. Without epigenetics, we will not be able to understand this, because genetics, we cannot, unlikely that we can change them in the near future, and we will not change them in these settings. And environment is very complicated, but it needs to have a way of changing us. And what can it change in us? Well, at the end, it's only epigenetics that might mediate this. There's not much else that could explain this. Some post-translational modification in some very general biophysical things. You can come up with a couple of candidates, but at the end, if we are all biological entities that work as the general theory is, there must be some change at the epigenetic levels. Otherwise, you cannot explain lasting change, I would say. Is the microphone on? Or? I can repeat.
Yeah, I will repeat. Thank you. So you said that adult neurogenesis was a driver of the brain individualization in these rodent models, right? So would you care to comment on that regarding human beings? Because there is, a question, how much adult neurogenesis do we have? And
yeah, there was one paper that somehow made it into Nature, and that is sort of in the face of the evidence of the field. And there's one more comment in cerebral cortex that goes into the same direction, but it's only a comment. All others agree pretty much with small changes that we have this phenomenon in the human brain. The studies are manifold, come with different methods. I think there is no real evidence. This one study based, and it's published in Nature, I must say, it's based on the fact that using a descriptive method that in the positive case would nobody have accepted as a positive evidence for neurogenesis. So the absence of replicating such a finding is interpreted as a proof of absence. And there you can quote Ullmann with absence of evidence is not evidence of absence. So this is a very strange thing. It was highly publicized. I think people like negative news, or I don't know. It does not make sense. It was a very poor study in many respects. There have been the solution to this, technical problems, all kinds of, I don't believe that paper. So I think it's pretty, so you can believe that you have new neurons. And perhaps you have grown a few in the, now in being exposed to enriched environment in this meeting. Huh? In the course of my talk and in this fabulous meeting.
Thank you, Gerd.
Hello everyone, my name is Sana Suri and I am a Lifebrain researcher from the University of Oxford, and I'll be chairing the next two talks. So it is my pleasure to welcome our next speaker, Dr. Nathalie Marchand.